The EV marker proteins CD9, CD63, and CD81 are transmembrane proteins. To develop antibodies that can detect the marker proteins on EVs with high sensitivity, we developed a DNA immunization method. The DNA immunization method is a technique in which host animals are directly immunized with DNA that encodes the antigen. The antigen protein is produced in the host animal, and antibodies that recognize the native form of the antigen can be produced efficiently. With the DNA immunization method, we made 6 clones of monoclonal antibodies, so you can choose the most suitable one for your application. Our antibodies can detect EV marker proteins with high sensitivity in non-reducing condition. We detected CD9 on the isolated EVs by Western blotting. Our antibody was able to detect CD9 with greater sensitivity than other companies' antibodies. The same experiment was also carried out for CD63. Our antibody is also more sensitive than those of other competitors. For CD81, also same results could be obtained. Our antibody is useful for scientists who need highly sensitive antibodies for EV marker proteins. Our PS affinity method is a novel technology to capture extracellular vesicles using TIM4 protein, which binds phosphatidylserine on the surface of EVs in a calcium ion-dependent manner. Based on this PS affinity method, PS capture exosome ELISA kit, streptavidin HRP, has been developed. EVs in culture supernatant released from COLO201 cells or iPS cells were measured by capturing them using TIM4, anti-CD9, CD63, or CD81 antibody. TIM4 could capture EVs more efficiently than other antibodies. Our ELISA kit based on the PS affinity method enables to measure EVs in sample with higher sensitivity. The ELISA kit shows a good linearity when performing dilution series of human serum and culture supernatant of COLO201 cell. We also provide EV detection kits using anti-human CD9, CD63, or CD81 monoclonal capture antibody. The assays are not influenced by the sample matrix, and a linear relationship between EV amounts in human serum and signal is observed. Those 3 ELISA kits also show good linearity in cell culture supernatant samples. PS capture exosome ELISA kit and CD9 capture human exosome ELISA kit were compared by using culture media containing 10% FPS spiked with human EVs. CD9 capture exosome ELISA kit shows high specificity for human EVs, whereas PS capture exosome ELISA kit detects human and bovine EVs in FBS. The performance of the new CD9 capture ELISA kit is not affected by the presence of FBS. We also developed exosome flow cytometry kit based on PS affinity method. You can capture EVs with magnetic beads conjugated TIM4 and detect them with fluorescent labeled anti-EV marker antibodies. The features of this kit are: enable highly sensitive qualitative analysis by flow cytometry, easy operation with magnetic beads, samples such as body flood or cell culture supernatant can be directly measured. Assay completes in 3 hours from isolation to staining. Using this kit, we conducted analysis of CD9, known as surface antigen of EVs, in serum or plasma samples. We have confirmed peak shift of fluorescence intensity in the sample, which was added PE-labeled anti-CD9 antibody.